Okay. Good afternoon, everybody. My name is Muthu Kumar Bhagwatyanan. I'm so happy to introduce our speaker today, Guy Coleman. He is a Fulbright Scholar as well as a PhD student at the University of Sydney in Western Australia, well, the New South Wales. I met Guy in 2013 when he was an undergraduate researcher at the University of Western Australia in Perth. I had an opportunity to visit Dr. Steve Powell's program where he was also working. We spent some time together going to the Western Australian wheat belt, collecting ryegrass seeds in wheat fields. Little that I knew that he's going to be a PhD student and a Fulbright scholar. Will be spending some time in my program at A&M. The world is very small. So anyway, so after his graduation, he was hired as a precision weed control scientist at the University of Sydney, particularly based at Narrabri, which is northwestern part of New South Wales. Um, Guy actually visited A&M about 2 years ago as he was attending a WSSA meeting here in the US. He spent a week here and he was pretty impressed with the A&M and all the facilities we have. And he went back and applied for a Fulbright fellowship. Sure enough, he got the fellowship and he started his PhD last year and started here at A&M as a Fulbright scholar in I believe it was early June. He will be here through December. At A&M, he has been very actively involved in greenhouse and field research projects looking at weed detection in agronomic crops, particularly palmer amaranth in cotton, as well as Italian ryegrass in wheat. So he has been collecting a lot of data. He's planning on presenting it at the upcoming ASA meeting. And Guy also he was very keen in learning wheat science and particularly applied wheat science here at Texas A&M. So he voluntarily took the wheat judging course this summer, which was not even required for him, but he registered and he did really well. And he went to the wheat contest, did pretty good over there too. So we are so happy to have him here and participate in the Fulbright program and do well here at A&M. Without any further ado, I want to pass it over to Guy. Guy, please take it away.
Thank you very much, Dr. Muthu. Very much appreciate that introduction. And as Dr. Muthu talked about, I'll be speaking kind of on those emerging technologies for weed control that I've found myself in, I guess, as my PhD and also over the past 3 years prior to that as a precision weed control scientist. But I thought before I could delve into some of the research, being an Australian and coming from a slightly different production system, I thought I'd take the time just to describe a little bit about Australia, the parts I've lived in. I've lived over a large portion of the country, I guess, and how that relates to agriculture and some of my research. So I grew up in the West Coast, the California, I guess, of Australia. Not quite, very different, but it's a city over on the far west, I guess, and quite distant to the rest of Australia. A beautiful place. This is a photo I took flying in I think one afternoon back in January before I came to the US. And it's got lovely beaches, a fantastic place to grow up. I also spent time in inland, so this is about 700 kilometres, I guess about 400 or 500 miles east of Perth in Kalgoorlie. It's a mining town in what you'd, I guess, classify as more of the outback Australia. So it's really hot, very flat. I guess it's technically a desert, but the amount of vegetation that's there is really something else. So I'm reluctant to call it a desert, I guess, but there's some beautiful scenery around Kalgoorlie as well. My dad also has a farm down on the southeast coast of Australia in a place called Gibson, which is, I guess, closer to the more famous Esperance. And that's where you see kangaroos lying on beaches with white sand and turquoise water. Really freezing water, though, because it's right off Antarctica. So it does make it pretty chilly for a dip. But that's my dad's farm from a couple of weeks ago. So he grows canola, and that's more of a sandy, very sandy soils down there. It only really opened up in the 1960s when they could get trace elements to support those sandy soils. But as you go further north towards Kalgoorlie from Esperance, you actually end up in nice clay loam soils. So there's a bit of a trade-off, I guess, between the reliable rainfall on the coast up towards the desert conditions further north, but better soils. Heading over east, as Muthu mentioned, the University of Sydney has a research facility up in Narrabri. This is about 7 hours north of Sydney. And that's where they have bred wheat for over, I think, 100 odd years, or since 1900, sorry, 60 odd years, I should say, I think. Basically, there's a whole heap of wheat breeding that goes on there. As you can see, a big block of canola. And then also these are the wheat trials in this center area here. That's all under linear irrigation, one big linear that goes back and forwards across that. And then also smaller platforms as well. And then finally ended up at the start of 2020, right before the pandemic hit, in southwest Sydney. So this is more of a market garden. I should say Narrabri is more of that self-mulching black vertisols. It's beautiful volcanic soils, very different to the country around Gibson and Esperance. And it doesn't need much nutrients or fertilizer, I guess, but it's mainly for cotton and sorghum, those irrigated crops. And in southwest Sydney, it's more of market gardens. It's on a floodplain, the Nepean River, and it's not quite typical wheat growing conditions, but we use it as a trial area for some of our data collection and some of our trials as well. And I thought the comparison, so that's Australia, there's Texas. To scale. So I think Texas fits about 3.5 times into Western Australia, and I think about 11 times into the whole of the country. So it's apologies to Tasmania for cutting them off, but it's a big country, but very variable soils and conditions. So this is the University of Sydney Weeds Research Team. I'm a PhD student there, but as mentioned, we used to be a precision weed control scientist and more of a research associate position. And the team is led by Dr. Michael Walsh, who's made a lot of moves, I guess, in the harvest weed seed control systems, integrated weed management, a range of areas. Now we're starting to shift into more precision weed control. But Assad, William, Eric, Linda, Jake, and Newman are all based around either Narrabri or Sydney. And we also have collaborations through Eric with the Australian Centre for Field Robotics, based in the CBD, the centre of Sydney. But we work on a whole range of things, as I'll describe. But it can come across as a bit of biology or agronomy. Cover crops is what Assad works with. Then we also delve into the precision weed control and alternative forms of control as well. So on the precision weed control front, these are kind of the areas we work in. We have a bit of work with robotics. So on the top left is a robot I built called Gordon in my lounge room, I guess, when I was a few years ago. And then we also worked in ginger for ginger weeding, site-specific weed control. That's an Ageris digital farmhand with one of our site-specific weed detectors on it. Data collection is another area. So we're looking at Weed AI as a platform I'll talk about later for open source images, data collection backpack systems to collect data more effectively. When I speak about data, I should say images. And also weed recognition as well. So how can we better detect weeds for a range of different methods? So bounding box or segmentation. And most of it is, or I guess all of it is ground-based. So I know some of the work here is the Bishwa and the team are doing. That's more aerial from drones, but we tend to be more on the ground with ground robots. And also alternative methods of weed control. So some laser weeding I've tested that I'll talk about, and things like the weed chipper, so site-specific tillage. How can you reduce, rather than till a whole field, you can just chip out the weeds instead. So for a bit of context around production systems in Australia that kind of guide, of course, what we do as researchers there. Predominantly, it's very large-scale production. So the average farm size in Australia for winter crops is over 10,500 acres. And so it's large areas. It's predominantly winter production. So winter for us is between May, often planting at the end of April. Some people might dry seed. So they go on before the rain in March with some canola varieties. And then they'll harvest often in around nowish, so October, even through to January in some of those Esperance South Coast areas where there's much higher humidity and rainfall. But that's typically cereals, oilseeds, legumes, and there's that strong winter period. And then after that, there's a fallow. So I'll try and maintain a weed-free, so there's no green bridge, I guess is what people call it, between that winter production system and the following year. So you're trying to reduce your carryover disease and insects across that period. So it's all about a lot of focus on weed control, removing weeds in crop and also in that fallow period. There is some summer season production too. So up near Narrabri, so just up where we were before. That's this area here and up into Queensland too is a large cotton, sorghum, and those summer beans, so soybeans for us, mung beans, those types of production as well. So that's a bit of a summary of Australia. We produce a lot of, I guess, a lot of grain, so most of it's exported. But there is a big focus on that winter season for the majority of the country. There are some summer crops in some places though, and most of it is dry land, not a whole lot irrigated, and the weed pressure is really often focused on that winter season, and that's why I tend to focus on weeds like annual ryegrass and wheat, or which is Lolium rigidum, multiflorum, or the Italian ryegrass here is I think also called annual ryegrass, but very similar, and I guess other broadleaf in cereal crop weeds such as wild radish or turnipweed, which is a close, looks similar anyway. But one thing Australian farmers face a lot of because of a shift to conservation agriculture that I should have mentioned. So there's tillage is not practiced a whole lot. So there's a lot of reliance on herbicides for weed control. And if any of you are familiar with herbicides and I guess herbicide resistance, you know that Australia is really leading the world in terms of the rate and severity of herbicide resistance spread. And some of the first cases were detected here, in Australia, and it's really developed since. So on the right, you can see these charts, I guess, frequencies. So green is susceptible populations that were sampled. Clethodim is a group 1 herbicide, so it controls grasses in wheat generally. And as you can see, when it was sampled in 2003, there's not a whole lot of resistance. 2010, you can see a substantial amount. And 2015, you can actually see some potentially shift back, but I'll leave that to the herbicide resistance people to talk about. But generally what you see is a trend towards a lot of resistance and often above 95%. So if you pick and this is for annual ryegrass as well, if you pick a variety or a plant up in a paddock, you'll often see that it's resistant to a range of group 1 herbicides at least. So that's a big cost to farmers. And it's because of a reliance on herbicides that has been driven by this conservation agriculture to preserve those really fragile soils we have in Australia that aren't heavy enough to really handle a lot of tillage. But of course, there's a lot of range of other methods as well of managing weeds that we're looking at. But that resistance does lead to a substantial increase in weed control cost. So upwards of $45 a hectare, and that's often due to opportunity costs where you can't actually plant the most, I guess, cost-effective crop. You have to plant something that fits in with that resistance that you've got in that paddock. And also the rotation or the herbicide uses you have to consider and factor into your system that factors into that $45 a hectare cost. And of course, then you've also got weed escapes too. So the cost of the actual weed competition and pressure on your crops because you aren't able to actually control them. And part of this challenge is that herbicide development is many hundreds of millions of dollars for a new herbicide. And so trying to get these new herbicides out there that might be able to challenge some of these resistant populations is difficult or non-existent in some regards. And so that's why we have this really big drive and interest from farmers and a range of different groups in developing alternatives. So how can we manage weeds, not just relying on herbicides because of resistance and a whole range of other things, but can we use alternative methods like perhaps lasers or targeted tillage or electrical weeding, a whole range of other methods, and of course cover crops and that whole integrated strategy to reduce that reliance and that cost then of herbicide resistance. And so that's what brought us to this first part of our journey, I guess, on this site-specific weed control story. And it was probably back in 2019, we're doing this work and looking at which technologies are available to us at the moment and why can't we use some and why can't we use others. And the deciding factor, I guess, because these are also varied, you have things like tillage or laser weeding, stamping, abrasive grit, microwaves, heating, targeted flaming, electrical weeding, even things like solar radiation. How do you compare all these technologies so diverse? Are they worthwhile investigating or not so much? And so what we came up with was rather than look at the potential cost of each of these methods, let's look at something like energy instead. So try and bring it down to a common factor to better determine what we should pursue down the track. And that was basically because there's no commercialization for many of these, and there's certainly no standard pricing on a weed basis. So that's what we did, decided to compare energy requirements for broadcast and site-specific basis and see what really fell out at the end. And this is what we got for broadcast control comparison. So down the bottom, you have all those different methods that I was mentioning. These are on, basically went through literature and found out how much, or the control, how much energy each of these used. On the left, you have the energy consumption in megajoules per hectare on a log scale from 1 up to, I think it's 100,000 and then 1 million megajoules per hectare. So megajoule being 1 million joules already. So blue is mechanical, and this, I should say, also includes both indirect and direct energy costs. So direct energy being the fuel used to drag the plow through the field, or the energy required to drive the tractor that's delivering the herbicide or drive the pumps, and all the indirect being the wearing of the plowshares, the requirement to produce that, the weed control component, I guess, not necessarily the carrier, but the weed control component, the required replacement. So that's why herbicides are quite high, is because whilst it's really easy to drive a sprayer of relatively low energy, the actual production of herbicide is relatively energy intensive. And as for things such as, the way, plastic mulch up on the right here, that's incredibly energy intensive, not necessarily in laying it down, but actually in producing the plastic to put over those rows and over those beds. And that's where you see these thermal methods in red. So the mulch is in that orange and herbicides in green. But those thermal methods, most of that energy is actually in the direct energy consumption. So it's the consumption of gas or propane, I guess, for those burners or for the heaters, for electrical weeding or any lamps or UV, I guess. That energy is always in the direct consumption. So that's the broadcast control. I guess it's what you'd expect. Tillage isn't that energy intensive really, whereas all your thermal methods are. And then herbicides sit somewhere in the middle. But the cost to the farmer of that energy is really in the cost of the herbicide, which are quite cheap. But then we decided, well, what if we could go down the site-specific weed control path? And the concept of site-specific weed control is really about targeting and treating the weeds instead of the paddock. So all those methods mentioned beforehand, they were all broadcast basis. So you drive the whole paddock and you're potentially spraying out herbicides over 100% of the paddock when there are weeds present on only 5%. So there's a 95% wastage there. But if we treat the weeds, not the paddock, you can, of course, reduce your inputs. And so there was some research showing it's between 40 and 90% normally on a typical basis. But of course, it's down to weed density. And there are additional weed control options as well. Because if you're reducing your input costs and therefore your herbicide use, there might be herbicides that are actually really expensive that do a really good job, but you can only apply them in very small amounts because of that cost. And then also we get the alternative methods. So things that were previously really illogical or irrelevant on these really large scale bases. So things like lasers or electrical weeding, maybe they can become relevant when used on a site specific basis. And so that's what we see with this chart here. So we have a whole range of different types of methods here. So concentrated solar, precise cutting, pulling, herbicides, again, as a check, chipping, water jets, stamping, microwaves, abrasive grit, electrocution, laser pyrolysis being the burning, and laser cutting being cutting. And again, estimated energy consumption on a megajoules per hectare. But if you look at the scale this time, it only goes up to 1,000. Because this was based on a weed density of about 5 plants per square meter and at the 2-leaf broadleaf stage. That's how we decided and paired things back. And mechanical, they're all quite low. And then thermal, we have wide ranges in values. So those bars representing the minimum and maximum values for each of those types. And so for electrical weeding, laser pyrolysis, and laser cutting, there's quite a wide range of energy consumption values because there's a wide range of types of lasers that you can use. And this also includes, which is a relatively non-significant, relatively important, I guess, aspect of site-specific control is the weed detection component. Because if you're running a GPU or a very powerful computer constantly to detect weeds, that consumes a fair bit of energy as well. It wasn't present in those other factors. And that's why they all tend to bottom out around the 14 megajoules per hectare. But of course, as things become more efficient, that could drop as well. So what we got out of this story is that lasers were really an option for us. So we looked at some of the emerging options. Actually, I'll just quickly summarize some of the thermal weed control. so how they work, and then I'll go on to lasers after that. But basically, for thermal weed control on a site-specific basis, we're looking at heat injury as the mode of action effectively. So it's a way of denaturing proteins, rupturing the cells, and then that has to occur between about 55 to 95 degrees, based on some research out of the 90s and early 2000s. And so we have these 4 or 5 areas, I think, that, I guess, again, modes of action, like kind of in inverted commas, these herbicides are often talked about in modes of action as well, which enzymes they target, or different parts of the plant. So here we've got flaming, which is a radiative or conductive heat transfer. Microwaves or dielectric heating, so vibration of the water molecules in the plant to heat up the surrounding tissue. Electrocution or electrical weeding is resistive heating. And then finally, lasers is more of a photon kinetic energy. So it's the actual incident energy of the photons hitting the plant, and again, increasing the temperature of that area. And so that's, I guess, where we landed with lasers. Not a whole lot of research done at the moment. And you can target that energy much more easily than, say, in the other methods as well. And that's kind of why we went down this path of laser weeding research. So on the targeted front, you can use lenses or mirrors to point that energy at any point around that you can target in that field of view of the camera, or just like you would engrave like a bit of wood or cut out a bit of metal using laser cutting. You can potentially do that for weeds as well. And so having that energy that's really targeted, but also flexible, and you can point it at the direction you need, that was a really big opportunity, I think, for that lasers still have. And I think we are still in very much the early days of this technology. And also they're relatively low energy. So what you consider a really high power laser is in the orders of 1,000, 2,000, 4,000 watts, or a few kilowatts. And you think about an average tractor power, which is in the range of many hundreds of kilowatts potentially, then you can power a lot of very big lasers that can control very large weeds. Hopefully, that's, I guess, what part of our research was looking at. And so that's why we went down this path, because while they are powerful in the laser community, if you've got a 4 kilowatt laser, in terms of agriculture, it's actually kind of in the ballpark of what people are using currently. And also they're portable, they're very small and lightweight, depending on the type of laser you get. So diode lasers are little kind of boxes you can get, and then they might have a fiber optic cable that leads it to a lens. As you can see in our system here, that orange cable is actually a fiber optic cable that goes back to a laser module back on top of the unit. And finally, they're no consumables. So as long as you have that electrical energy, you have those kilowatts available, then you can run a laser. You don't have to worry about, say, abrasive grit. You need the high pressure water in the grit. Things like inflaming, you need the propane or those kinds of consumables. Lasers are great because you just can, as long as you've got that energy and that power source, you can drive them. So down this path of laser weeding, we wanted to work out, well, are they relevant for weed control in large scale systems? And it was mainly to look at if there is what is the efficacy of lasers. There hasn't been a whole lot of research done on weeds that are relevant to Australian production systems and large scale areas. So we looked at annual ryegrass, so Lolium rigidum, and so rigid ryegrass, and turnipweed, the Rhipistrum rugosum. And these were 2 key weeds up in the Narrabri region for the winter crop. We want to look at how much energy was required for control, and also do these different species and different growth stages of the weeds actually influence the control energy. So say if you're going at the late stage of the crop, how much energy you're going to need to control those large weed escapes, or if you go in the early stage, how much do you need for those 2 or 3 leaf stage weeds? And so this was a high energy testing. So it was after a bit of preliminary work we did, and we're using a 25 watt, so just about a light globe, I guess, a few LED light bulbs, a 975 nanometer wavelength, which is in the near infrared. And we looked at annual ryegrass, rigid ryegrass, and turnipweed. And we used the energy density, so which is the density of energy on the spot that it contacts the plant, as a way of standardizing across different treatment durations. And so basically that's the power by the time. So that's the energy consumed, and then divided by the area of that spot. So you end up with a joules per square millimeter unit. And that was over 4 growth stages. And so we targeted these different energy densities. So we had 6 different energy densities across all those growth stages. And we used up to a 305.6 joules per square millimeter, which corresponded to about a 4-minute treatment, which is really long. But the idea being you can deliver that energy much more quickly if you have much more powerful laser. So because 25 watts is really quite small. And this is what it looks like. So this is a laser infrared. You can see it kind of overcoming the infrared filter on my camera. And you can see the weed in the center getting burned and then kind of shriveling up and changing. And so that was for a 30-second treatment. It's being sped up a little bit. And that was 38.1 joules per square millimeter. And you can see there's discoloration in the center there where those cells have been ruptured and the cell contents are leaking out. And this is what the damage looks like afterwards. So this is on the largest turnip weed we treated. So that's 150 seconds. You can see the burning in the center. And there's also a ring of damage around that where it hasn't burned, because burning is well past the control stage. So burning is almost a waste of energy. And as you can see, similar for the ryegrass there. And this is what the results look like for the rigid ryegrass. It was fairly similar for the turnip weed. I'll just stay with ryegrass in the interest of time. But basically, you can see that for the youngest 2 growth stages, we had fairly good control. So when this is the biomass, the percent of untreated control, so everything starts at 100%. And then as you control it, or as you kill most of the plant or it doesn't regrow, then you'll see these values should come down. And then on the bottom here is the laser energy dose. So as you move from the left over to the right, you'll increase that energy dose and therefore hopefully the chance of controlling that weed. And where you see these values come down, it was about sort of 9 or 10 joules per square millimeter, which corresponded to about, I think, 15 to 30 seconds to double check. And then for the second time or second growth stage, which I think was about the early tillering, then you could see it was more tolerant to the laser because, I guess, of that larger biomass you'd see in those the growing point. And then out to the fourth, or the largest growth stage, so it was flowering for the turnip weed, and it was very late tillering, almost flowering for the ryegrass. You can see there was no control, but there was some suppression of those plants. And I guess what's also interesting is that the ryegrass was more for the first 3 growth stages. It was more, I guess, susceptible to the laser than the turnip weed. So I think because of the structure, I guess, the turnip weed has some hairs on the leaves that may have influenced that. And this is the visual depiction of those results. So for the earliest growth stages, you see fairly good control across all those energies tested. And then as you go up, as the plants get bigger, you expect them to survive more readily. And you can see where it's controlled half of the plant, the other half has survived. And so it's given us the idea that a single spot size that's 5 millimeters wide is probably too small. If you can try and scan a plant or do other things with it, then you might be able to have better luck at controlling that entire plant rather than just half of it. That'd be more efficient with the energy that's being delivered. So site-specific weed control, so delivering these things on an individual basis is enabling us to use these alternative methods. And what we saw was that lasers can control the 7-leaf annual ryegrass and also turnip weed. And what is probably more advantageous for us is to look at better ways of delivering that energy rather than keeping and increasing it further. So can we find better lasers that are either more powerful and deliver that energy faster and over a larger area? So you end up with a better efficiency overall, or the better ways of using lenses or optics that can improve that delivery efficiency too. Or we could just go with the Star Wars method of zapping a turnip weed that way. Spent way too much time in that GIF. But as I mentioned previously, site-specific weed control is really the backbone of that. It's been doing a lot of heavy lifting. There's a lot of assumptions with these laser systems that we can have a site-specific weed control rate. Because if we can't even detect a weed, well, there's no point looking at lasers because you're not going to laser a whole paddock. And it's a good idea, right? But how do we do it? And that's where weed recognition really fits in. And that's all about trying to work out where the weeds are and detecting them. So then you can go out there and actually control them. And I guess this is a good example of potentially how computers are not very good at detecting weeds. Because I reckon who here has any idea what's in that paddock? Any ideas on the weed or the crop or what stage it's in? This is a field in Australia. Any guesses? Yeah, see, wheat's been harvested. Good guess, Muthu. Any Kenizer? Oh yeah, good guess. So that's look, so Muthu's been trained a little bit, perhaps. Like he's maybe a weakly trained machine learning model. He's been to Australia a few times, but Microsoft did a terrible job because they said a field of brown grass. And I guess as humans, like if we look at that, even probably a bit up close and when it's not so blurry, you can see there's stubble there. You can see there's sparse plants growing in between. And if you're walking in that field, you could probably get a fairly good understanding of what might have been there, or even very quickly, someone could tell you that in a sentence or two what's happened here, and you'd be able to see that that happen. So for humans, it's quite an easy process to learn what a weed is and what isn't. And this is probably quite tricky because it's a very dense canopy of hairy fleabane or Kenizer, I think it's Amaranthus. But that's a very common fallow weed in Australia. So this would be post-harvest, probably around December, I'm guessing. But if we want to train a computer to do this, it's slightly more time-consuming, I guess, and it requires a lot more data and more time in trying to teach the computer or let the computer teach itself, I guess, what's important in those images. And so to give a bit of context for it, I thought I'd just quickly go through some of the key terms like artificial intelligence or machine learning, deep learning, those kinds of things. So AI, artificial intelligence, is basically systems that act like humans. So they're things that, inverted commas, it's things that do things a little bit more intelligently, I guess. So it's rather than your washing machine just going the same program for every cycle, it might have a sensor on it that changes the pressure or the temperature or the duration based on water quality. There's no machine learning involved there. It's just a sensor that has a bit of light passing through the water, and if the light's more interrupted or something, then it'll go longer. So that's artificial intelligence, I guess. It's not just some set program or set system. It actually has a little bit of intelligence to it. Machine learning is that, I guess, it fits within artificial intelligence. What it is that it can actually learn itself. So you don't have to tell it what to look for. In the case of the dishwasher, you don't have to say, look for the light beam and the way it's interrupted. You can say, here's the output. This is what the results are. This is the answers. And then it will learn from those answers what it should look for and what it should do, what the inputs are. And so what it, I guess, by definition, is it's learning without any explicit programming. You give the answers, and it works out the process of getting there. So when it sees something in the future, it can do that same process again and give you some form of prediction. And so what deep learning is within machine learning, but it's just a much more, I guess, a whole series of more complex or fairly simple algorithms, actually. So linear algebra. There are many millions and millions of those simple equations that are combined together over large, what are called neural networks, that they can then represent much more complex systems. So machine learning can be quite simple. It can just be a few different things connected to each other. It has a feedback system, whereas deep learning might have many millions, even billions of parameters that can be trained and learned on large data sets. And what we really sit across in the weed detection space is what's called computer vision. And so computer vision is simply just trying to understand what's in an image using a computer. So if you have an image of those weeds in that crop, then you're trying to understand what the weeds are, what the crop was, where you can control and where you can target those. We're trying to derive context from that. And humans are really good at seeing an image and immediately understanding what's in it and getting context out of it. But for computers, it does take a little bit of time for them to learn and for you to work out how to best target the training data. So that's how everything fits. And I tend to work in deep learning. So with those artificial neural networks or convolutional neural networks that do most of the heavy lifting for you. So they extract the features you want and then they train and you basically just press go and it works the rest out. And then what it comes down to really is pattern recognition. And there's a journal called Pattern Recognition that's all about deep learning and machine learning. Because at the end of the day, computers, they just see numbers. They don't see any of the meaningful things that we see. They just get a series of numbers. Now looking for patterns between those numbers at either the primary level, so they just see the literal number, or maybe they might interpret it a bit and apply a few equations to to get those more lower level or tertiary relationships, or many millions of layers deep into those relationships, to try and understand what's in there. But this is an image of ginger and a couple of weeds. And these are actually the numbers that are behind that image at a very low, obviously, resolution. So for an RGB image, you have red, green, and blue channels. Each channel usually has values from 0 to 255. I've just taken the green channel here. And each of these represents a number from, like an intensity of light on that green channel that's been detected by the camera sensor. And that's what the algorithm gets to train and to develop an understanding of those relationships between what a weed is and what a ginger leaf is or isn't. And that's what we're dealing with, I guess. And it's a lot more, I guess, that's why you need so many images and so much data to try and develop and understand these subtle relationships between leaves of slightly different colors or shapes than, say, a car and a horse, or things that are much more distinct. But what's exciting about deep learning and why we tend to focus on it is that the more data you give it, it tends to continue to get better. Whereas older machine learning algorithms, so that one circle out, they tended to plateau with the more data you gave them. And also, you could only, well, back then, the idea was you had to extract the features for it. So you told it what to look for, you told it to look for the green, or you told it to look for edges or different textures, and then it ran on what you told it to look for. Whereas some of these deep learning networks with convolutional filters, or things that extract relationships or colors or patterns for you, as you give it more data, they do tend to get better. And this is from Andrew Ng. He's a professor at Stanford. He's done some amazing things for machine learning and education as well. He's got lots of free courses on YouTube. So if you're ever interested in machine learning, he's certainly a guy to follow and to listen to, and I guess learn from lots of his videos. But that's why there's this hype, I guess, because that performance and the development of performance hasn't been seen with previous algorithms. Not that machine learning or deep learning, I should say, can solve everything. It's just that there's this relationship, I guess, that as you give them more data, it continues to improve. And so what we get out of these models then, so we've done all the training and all that, and this is what we end up with. So as you can see, there are generally 4 different types of weed detection or recognition that we get. And classification is the broadest, so it just tells us whether or not a species or an object is present in an image or absent. You don't know where it is, it's just that in this frame, in this image, there is something there. Then you have object detection, which I'll show later, and that's where you have 4 different, in this case, turnip weeds, and you have, where the location of each of those turnip weeds and how many of the turnip weed there are too. So that gives you more information there for more targeted control potentially. Then you have at the pixel level, so as you had red, green, and blue intensities for every single pixel in those images, at that same level, each pixel, you can classify those pixels into the positive class, so the weed you're after, or the weed you're not. And that's what semantic segmentation is. But again, you don't know how many weeds there are, you just know that there are X many pixels that belong to this weed class. You can use some sort of other computer vision methods to extract and differentiate, say, this cluster from this cluster, but you can't actually count which plants are which. Whereas then we get to instance segmentation, and we get some really advanced, I guess, outputs from these models. So we know now that there are 4 different turnip weeds there, and we know how many pixels and which pixels belong to each of those instances, or each of those weeds, each of those plants, each of those turnip weeds. And this potentially, or these kind of level of detection is what we might need for, say, laser weeding in the future. So we can scan that whole plant and potentially fire a laser across the whole thing rather than just the growing point. And there are other methods of detection, so key point detection, or even you could detect, say, the growing point, or you could detect features like insects or disease spots. In this case, this is all actually drawn manually, I've just as a demonstration, but you can just draw a box around each individual weed and train it on weed detection as opposed to like spot or growing point detection. But that process of training it and developing the annotation data is incredibly intensive and frustrating and very boring. So this is me doing some ryegrass annotation, which is the same data set in the previous image. And it's a very painful process. So I've just recently, in the next model I'll show, trained or annotated with the help of Sarah, about 3,000 instances of palmer. And so you have to collect those images, you have to go over each image and then draw a box around every palmer amaranth, or every ryegrass growing point, or every weed you're after. And it gets pretty tedious pretty quickly, and it costs a lot of time and I guess money in labor. So that's the reality, right? Like it's really exciting and lots of hype behind lots of these things, but the reality is, at the end of the day, you have to at least start with labeling all these images for the weeds you're looking for. And so that kind of brings us to my research now. And we're looking at these development areas of how we can access large image data sets that are annotated, they're diverse, they're weedy, so to have all the weeds we want and the conditions we want. Can we search and kind of find those conditions and those weeds we're after? And then I'm also looking at if the relationship is, say, between the plant appearance and the performance of the algorithm, what's the relationship there? So as the weed matures, so say if we highlight these two palmers here, they're both palmer amaranth, but they look vastly different. So how do you best train an algorithm to detect those two plants consistently whenever you want? And of course, a really important area of research is reducing the annotation requirements. So can we reduce the efforts of those bounding boxes and cut back where we can and just focus on what's really required? I know like Matt and Bishwa and Alberto, they're working hard as well on that, on different methods of capturing data in semi-field or field environments that can improve that too. And so things like unsupervised algorithms, where you give it a whole bunch of data and it looks for things that are different, is that a new method that we can try to reduce that workload in different environments? And synthetic data as well. So you make up your own images and just splice in weeds where you want. That's, again, another opportunity that might reduce some of that workload. And then finally, what we're looking at is open source hardware too. So what's the opportunity to bring on people from around the world in semi-related or even unrelated fields that are interested in agriculture and weed science to contribute their knowledge to open source software, hardware, that kind of development. And I know, like myself, I've benefited a lot from all the free tutorials that people like Andrew Eng have provided. And can we give those a bit of an agricultural or weed science flavor and get other people to learn about machine learning and robotics, but through the lens of weed science and agriculture too? And that's kind of the areas of development that I'm looking at. So firstly, on the open source data front, this is a platform I've helped develop called Weed AI. So that's weedai.sydney.edu.au. You can check it out on your phone. And basically, it's just a way of bringing all these images together, standardizing reporting, so everyone can upload and download and develop this community around data, image data, and annotated weed data sets. So that's on that data front. Then on the impact of plant morphology, this is some of the work I've been doing while I've been at Texas A&M. So it's how do we work out how crop and weed growth stages can influence detection performance. So what I went through with this work was basically capturing images of palmer amaranth using this technique here. So you have a backpack with a camera out the side, walk around the field in the hot sun and sweat onto the keyboard and it breaks. But it's how do you capture enough images and annotate it enough? So you can either do multi-class, so you get information on different growth stages, or single class, so you just have a palmer amaranth class. Which ones perform better? So I'm just at the moment of training models up for that. And so I used 8 different growth stages. So from seedling, where it has just 2 leaves and cotyledons, 3 to 6 leaf, over 6 leaf, where the palmer where it's developing the inflorescence, where it's flowering. And then also palmer amaranth can have a spreading or like more of a prostrate growth habit. And so by looking at these spreading, I guess, phenotypes, then you can potentially separate them out and maybe improve the model that way. And so they have the 8 different model classes that I gave it. And this is the performance, fairly hot off the press. So as you can see, it's what we have here is the precision and recall in the black and the white bars you can see. And it was trained on what's known as the YOLO algorithm, or you only look once. It's the state-of-the-art algorithm, version 5, which is uncomfortably easy to train, because if you have the data, you can develop a detector of some sorts. And they have different size architectures. So S, which is small, and M for medium architectures, and then at 2 different resolutions. Obviously, the more higher resolution you give it, the more detail it can pick up potentially. So that was a way of working out if we could pick up small weeds. And so then tested on multi-class, so there's 8 different classes, and also the single class groups as well. I haven't got any data for that at the moment, but just finished training before I came down, and I can talk about that shortly. But basically for the multi-class, so this is across all 8 different classes. So Palmer Amaranth 1 is that seedling stage, all the way up to SPA or spreading Palmer Amaranth 3, which is the flowering spreading Palmer Amaranth. You can see that precision, which is the ability for that algorithm to, when it makes the detection, is that detection correct? So it said, all right, this is Palmer. And so for across the whole algorithm, it's about 54, 55%. That's saying every time it makes a detection, that detection is actually a correct classification. Then we have recall, which is the ability of the algorithm to, out of all the weeds that are present in the training data, how many has it actually correctly identified? So it's probably most important for our farmers because they often they'd rather overspray and spray the areas and get false positives than miss weeds that then germinate and go and set seed. So recall is fairly important. And so just initially there's about 60% across the algorithm tested. This is very early data, so I hope to tune it more. But what's more interesting, I guess, is you can see different classes perform substantially differently. And it's probably due to some relationship with the sample size, and also that these classes of inflorescence development and flowering are also quite similar to each other. So the next stage of this research is grouping them together and then trying to minimize or make those classes really clear, so the algorithm is more confident on what to detect. Same as these 2 classes here, they're fairly similar. So it didn't, I guess, if you group them, you might see performance increases. And so this is a confusion matrix. And so what it's showing down the bottom is the class that's the correct class. So everything going up is the correct class this way. And this is what it predicted. So a perfect algorithm, you'd see a 1.0 on this diagonal. These predict everything as PA1. And so this gives us a bit better understanding of, okay, so a Palmer Amaranth 2, so that's the 2 or 3 to 6 leaf stage, it's actually picking up lots of those as Palmer Amaranth 1s. So there's a bit of confusion there. And also, if we go to Palmer Amaranth 4, it's some of those are ending up as Palmer Amaranth 5s and Palmer Amaranth 3s, when the actual class is actually quite low. So for that class, maybe that does need to be moved into either the Palmer Amaranth 5 class or some of the other ones that might have been mislabeled potentially into the Palmer Amaranth 3s. It gives us a better way of understanding how these growth stages are impacting on detection performance. And then these ones down the bottom, they're actually one big clump. So all those spreading Palmer Amaranths, it's confusing a fair few of them into the other. And that's what we can learn from something like this. And what I said with that 1 class algorithm is that it actually performed quite well. But I do need to check how or where the detections are missing. And often it's because it's got a lot more data to train on, and so it can perform better. But I guess that's my work, we're looking at understanding more about how these detections work. And that's what it looks like. So that's it, picking up Palmer Amaranths, different growth stages over at the cotton on the farm. So what's interesting, I guess, as well, is you can now get different growth stage information and then potentially make decisions on how to spray or control that weed based on its growth stage. And so finally, this is the open source system we just released called the Open Weed Locator. And this will kind of bring everything together. So it's the detection, it's the hardware, it's the actuation, it's the weed control. And so all you have to do is just plug in, say, a solenoid for a spot sprayer or a targeted tillage. And at the moment, it's just green on brown. So it's that fallow weed control that is currently detecting. So it just detects green. It does an okay job of it. So we've tested across about 7 fields in New South Wales. So just around near Sydney. And it performs averagely, I guess. But when you use it on, say, a single field, the performance goes up quite substantially. And that's the issue with color-based algorithms, which deep learning, I guess, came around to try and solve. They're not very good at generalizing, whereas deep learning can. And so while we're starting simple, the idea is that people can contribute to this, build their own, learn a bit about weed science and weed detection and robotics through the platform too. So hopefully it's an educational and practical tool that farmers can use or be interested in and maybe might improve adoption or look at those avenues of encouraging the understanding of the technology. But it's something I'd hope to test with green on green algorithms as well. We might need to update the hardware because it is just a Raspberry Pi, which is a very simple and low cost embedded computer. If you want to check it out, it's Open Weed Locator. If you Google that, it should pop up on GitHub. And that's it on that robot you saw at the start, so the Agaris digital farmhand. So I was just doing a fence line. It was a couple of weeks before I came to the US. The robot is a company in Narrabri in New South Wales. So about 7 hours north of Sydney. Not in Texas, unfortunately. Yeah, so that's just the University of Sydney's farm they have over that way. Yeah, finally, I thought I'd just mention the Fulbright Scholarship. So very, very fortunate to receive this and come over to the US. Very appreciative of the time that Muthu's taken and helped me submit the application, the help I've had from people here. And it's really a bilateral program. It was started after World War II by J. William Fulbright. And it really is about educational and cultural exchange. So as much as it is about the research, it's also about fostering engagement between those 160 other countries and the US. So I get to learn about the culture here in Texas and the United States. And then people from the US can come to Australia or other countries around the world and develop research skills and collaboration, and it will hopefully last for a long time. And it's all about developing that mutual understanding. And so would very much encourage people to apply if they're at that stage of their in the US, if you're recently graduated or about to graduate from your undergraduate degree, or if you're a master's or doctoral candidate, then you can also apply for it. I think elsewhere around the world, so for Australia, you can also apply as, say, a visiting chair or a postdoctoral researcher. There's a whole range of other opportunities. But I just noticed on the US Fulbright site that it was just those two groups. But yeah, make sure if you're in the US, check out the US version of the Fulbright, or if you're elsewhere around the world, then check out your country's version of it, because it's certainly a fantastic program and something I'll look back on fondly for sure. Because it's been a great 3 or 4 months now, I think, at Texas A&M, and I've had the great opportunities to visit Mississippi State for the weeds contest with Muthu and the crew, chip weeds in 100 degrees, and also helped hemp harvest and learn about things like hemp and cover crops that I never would have potentially really had access to elsewhere, and just working out how other people do things around the world is really, really invaluable. So very appreciative for the experience, and looking forward to the last, I guess, couple of months here and making the most of my time. So thank you very much. Thanks to Fulbright and Texas A&M and Sydney Uni and everyone, I guess, involved that's helped me with this research. So any questions, feel free to ask them. Get in the front.
Yeah, I have a question related to the lasers and the applications. I think so. Basically, your main application using lasers in this context would be like killing those non-desirable plants or so. And I got a bit lost on the description, because in some places you use energy, in some other places you were using power, in some other places you were using energy per unit time, per unit area.
Yep.
But it come to me like intensity. And then I got a bit lost on those parameters that are like totally different parameters that you use to judge what you need. I understand that what you are using is 1075 nanometer wavelength.
No, 975, yeah. Near infrared.
975. Okay, this one is like very strong absorption of water, right?
Yeah, strong reflectance from plants, I think, and strong absorption from water.
Okay, because I think this you also have like water absorption quite strongly around
like
975.
That's probably about right, I think.
It's around like 400. It's a little bit further than 950.
Because we are using this and we have problems with like for quantum thermometry, but I think it's 975 that you get that the plant absorb. But okay, in any case, so my question is all those parameters, and I'm very surprised, you know, like I develop lasers.
Okay.
So and in addition to these, one thing that where you kind of take care is like Is it a continuous wave laser? Is it a picosecond laser? Is it a femtosecond laser? Because with the femtosecond laser, you can produce in a very short time, maybe what New York would use like in 1 day, right?
Yep.
Maybe you might be able to destroy things faster. But there are also like, for me, it's like, I'm not sure, I mean, in terms of cost, right? And in terms of applying the technology, but there is a fundamental issue too, that like the safety conditions, right? If you shoot a femtosecond laser down there, it's like, well, I don't know. I mean, definitely no people should be around, right? Because this is it's like these surgeons that at this level, still you can use it like scalpels, right?
Yeah.
But at that level, so it's still like, I mean, I'm excited that lasers are going to be useful maybe for something like that, but I have the impression that, wow, it's like, and I think you have just been used like continuous wave laser, right? Because you mentioned you were using just this, but okay, I read you like.
All right, yeah, so yeah, continuous wave laser is what we used. It was a diode laser. We did try a nanosecond laser, so 20 nanosecond pulses, 1.5 kilowatt average, but it was at I think 1,075, 1,050 nanometers. That was different, and that didn't work very well, even though it's very high intensity pulses. So basically, pulse lasers, I hope I'll try and describe, let me know, is where you deliver the energy in a very short, sharp pulse duration. And those pulses happen at a certain frequency. So you might happen at 20 or 60 hertz. And so while the average energy is quite low, the individual energy of each of those pulses is very high. And so you can potentially get very large energy deliveries over very short periods. But anyway, we tried that, and I think it's more of a wavelength thing.
Wavelength thing,
yeah.
Right, exactly, because you have to target so that the energy will be like absorbed or
exactly, yeah.
Because I think you want to destroy, so this means like, yeah.
For sure, yeah. So
it is interesting.
So yeah, that's what we're trying to research as well, is like if you apply a red or a blue laser, which is strongly absorbed by plants, maybe they'll be better because the wavelength, the energy is going to be more absorbed, whereas 975 nanometers is going to be strongly reflected. So those sorts of questions too, not necessarily just the application.
It's still 975 and some absorption there, but it's all relative, right?
Yeah.
But you can still look for a region where you get stronger water absorption and kill the whole thing. Mm-hmm.
Yep.
Okay.
Yeah, I'll have to chat to you after.
Yeah.
Up the back.
Yeah, I have a question about the picture you probably. So I'm just wondering about the beta location.
Yep.
What kind of resolution?
Was that the ginger? Was that one?
Resolution.
Oh, so the images I'm using at the moment are 3200 by 2208. So they're 7.2 megapixels roughly, but they're downscaled to 640 by 640 for the YOLO V5 or 1280 by 1280. And it's hard to say like definitively if it performed better, but from what the early results I saw, the high resolution was better. Because as you'd expect, those really tiny cotyledon plants weren't just single pixels. They might have been multiple pixels instead. So that's why I'd assume that the high resolution performed better.